Hello everyone, welcome to VLearn and welcome back to the class Molecular Basis of Inheritances. In the last class of Molecular Basis of Inheritances, we talked about the process of the DNA replication, which is basically semi-conservative in nature. So we proved that by Michelson and Sethel experiment, the DNA replication is basically semi-conservative in nature. In today's class, we are basically going to continue the DNA replication, and in today's class, basically we are going to study about the mechanism of the DNA replication. So if we talk about the DNA replication, DNA is a long stretch of the base pair. In a single eukaryotic cell, it is around 2.0 meter. Remember, we calculate the length of the DNA and it was found to be around 2.0 meter in eukaryotes. So it is not a single small stretch, it is a long stretch of DNA. So basically, the DNA replication, not start at any point. It start at some specific point, and that specific point at which DNA replication initiates is known as ori, or it is known as origin of initiation or origin of replication. So it is known as origin of initiation or origin of replication. Initiation, or it is also known as origin of replication. If we talk about this origin of initiation, this origin of initiation is different in prokaryote and it is different in eukaryote. So if we talk about prokaryote, PK means prokaryote. Prokaryote means bacterial cells such as E. coli cell. In case of prokaryote, this origin of initiation, that means replication, the process of the replication only starts at a single point. So in case of prokaryote, the process of replication process occur at single point. Replication occur at a single point. Therefore, it is also called as monorepliconic. Therefore, it is also known as monorepliconic in nature. Monorepliconic. Because the process starts at single point, therefore it is monorepliconic. Therefore, it is known as monorepliconic. But if we talk about eukaryotes, the process of the DNA replication occur at many points in eukaryotes at a single time. Let us say this is the DNA in case of eukaryote. The process of the replication, the origin of initiation, occurs at many points. That means eukaryote have many ori. That means the process of replication occur at many points at single time. Therefore, the eukaryotic DNA is basically multirepliconic. So the process of DNA replication in prokaryote is basically monorepliconic, and in eukaryote it is multirepliconic. From exam point of view, we are basically going to study about the prokaryotic DNA replication. We will also discuss some points about eukaryotic DNA replication, but will not go into detail. So in your syllabus, basically it is all about prokaryotic DNA replication. So if we talk about the prokaryotic DNA replication in prokaryotes such as E. coli cell, replication in prokaryotic cell. So the prokaryotic cell की यदि हम बात करें तो the process of the prokaryotic cell, we know that it is monorepliconic. That means replication would occur at only single site. So if we talk about the replication in prokaryote, the prokaryotic DNA is double-stranded circular DNA. Let us say this is our DNA. And at this point of time, the origin of replication initiate. This is called the ori. And at this point, the origin of replication starts, and it starts in both the sides. That means the origin of replication occurs both the sides. This means that the DNA replication in prokaryote is basically bidirectional. So DNA replication is monorepliconic and bidirectional in case of prokaryotes. The point at which this replication starts is known as ori, or origin of initiation or origin of replication. And the point at which this terminates, it is what we call the termination site or the ter site. So there are few termination sites at the opposite end of the ori in case of prokaryotes. So this is the ori. So let's start this process of replication. By now we know that the process of the DNA replication is monorepliconic and bidirectional in case of prokaryote. Let's start with DNA replication in prokaryotes. So if we talk about the process of the DNA replication, the process of the DNA replication starts at ori. If we talk about ori, we are basically particularly calling about E. coli cell. This origin of initiation region is around 245 base pair long. So let us say this is the part of the ori. I am basically zooming in this origin initiation part. So this is, let us say, the region which we called ori region. If we have a look at the ori region, let us say this is the 5' DNA, this is the 3' DNA, this is 3' DNA, and this is 5' end of the DNA. This is only the DNA of ori region, this part. This is the only this part of the DNA. In this part of the DNA, there are some sequences. There are some sequences which are basically conserved in nature. These sequences here are what we call Mer sequences. There are some 13 Mer sequences and there are some 9 Mer sequences. There are 13 Mer sequences and there are some 9 Mer sequences. Let us say this green part here is what we call 13 Mer sequences. And this pink part here is what we call 9 Mer sequences. This ori region, if we talk about a prokaryotic DNA is basically GC rich. GC rich DNA. That means the most of the DNA will be containing a more amount of G and C, because it provides the stability. But as its name suggests, in the case of ori, the ori is the part of the DNA at which origin of replication, that means replication, starts. So this ori region is basically not ori GC rich. This is basically AT rich. Why it is AT rich? Because basically to start the process of DNA replication, we are going to separate these strands of the DNA. We are basically going to separate these strands of the DNA. And for separating these strands of DNA, we are going to break hydrogen bonds in between them. So if we are going to start it with a region of GC, high amount of energy will be required. But if it is AT rich, because A and T are bonded to each other with only double bond, low amount of energy will be required. So in order to start the process of the replication, the The ORI region is basically AT rich. That means adenine and thymine का जो amount है that will be present in more quantity compared to the rest of the DNA which is GC rich, because अभी हमको process of replication start करना है. ठीक है, इन दोनों स्ट्रैंड्स को सेपरेट करना है एंड सेपरेट करने के लिए ऐसा रीजन होना चाहिए जिसको हम इजीली सेपरेट कर सके. Therefore this region is basically AT rich DNA. Now let's come to the another point, 13 mer and 9 mer sequences. That means these are basically 13 base pair sequence and these are basically 9 base pair sequence. These Mer sequences are basically conserved in nature. What do you mean by conserved in nature? It is also known as 13 box or 9 box. So the ORI sequences and these sequences, which are what we call 13 Mer sequences and 9 Mer sequences, the process of going to start at these points only. And these sequences are conserved in nature. What do you mean by conserved in nature? Conserved in nature means these sequences, these 13 base pair sequences or these 9 base pair sequences will not be present anywhere else on the DNA. These 13 base pair sequences is conserved. That means if we mutate this part of the DNA, if we are going to mutate this part of DNA, there will be no replication or there will be small amount of DNA replication, because this is the part of the DNA by which the enzymes are going to locate the site of the DNA replication. Without these conserved sequences, the enzyme such as DNA helicase will not recognize where to start the DNA replication, because the stretch of the DNA is too long. तो बहुत बड़ा स्ट्रेच है डीएनए का और हमको स्टार्ट करना है बिल्कुल छोटे से पॉइंट से रेप्लिकेशन का प्रोसेस, बिल्कुल 245 बेस पेयर की सीक्वेंसेस से. एंड हेलिकेज जो है वो यहीं पे आगे बाइंड करेगा, वो कहीं और बाइंड नहीं कर सकता. ठीक है, तो दैट मीन्स बिकॉज दीज सीक्वेंसेस आर कंजर्व्ड इन नेचर. कंजर्व्ड इन नेचर मतलब ऐसी सीक्वेंसेस तुमको और कहीं पे नहीं मिलेगी. 13 मर की सीक्वेंस, दैट मीन्स जो 13 मर की सीक्वेंस एग्जैक्टली यहाँ पे है वही सीक्वेंस यहाँ पे है. 9 बेस पेयर्स की जो सीक्वेंस यहाँ पे है, दैट सेम सीक्वेंस इज प्रेजेंट ऑन दिस पार्ट ऑफ द डीएनए. So these are highly conserved sequences, and if these conserved sequences, if any conserved sequences of DNA undergoes mutation, it will fold that process. That means यदि आपने origin of initiation के इन sequences के अंदर कोई mutation cause कर दी है, that means DNA helicase will not bind to this part of DNA, and DNA हेलिकेज विल नॉट एबल टू रिकॉग्नाइज कि मेरे को कहां से डीएनए रेप्लिकेशन प्रोसेस स्टार्ट करना है. तो इससे डीएनए रेप्लिकेशन का जो प्रोसेस है दैट विल बी होल्टेड. देयर विल बी मिनिमम रेट ऑफ रेप्लिकेशन और देयर विल बी नो रेट ऑफ रेप्लिकेशन. तो बेसिकली ये जो सीक्वेंसेस है यहीं से क्या होना है, डीएनए रेप्लिकेशन की प्रोसेस को स्टार्ट होना है. ऑलराइट, initiation, this is the initiation part. This is just the beginning of the initiation part. Now let's go to the biochemical part. If we talk about this stretch of DNA, let us consider this is the stretch of DNA. This is the 5' end of the DNA. This is the 3' end of the DNA. This is 3' end of the DNA. This is the 5' end of the DNA. And this is the region where basically the replication is going to be started. These base pairs, these two strands of the DNA are basically connected to each other by hydrogen bonding. So these are the hydrogen, these white lines I am representing is the hydrogen bondings between the two strands of the DNA. In order to start the process of the replication, we have to break these hydrogen bonds. So we need breakage of the hydrogen bond. Breakage of hydrogen bond. In order to break one hydrogen bond, we need 2 ATP. For your general information, this is the amount of energy we are going to need in order to break the one hydrogen bond. For breaking hydrogen bond, there is an enzyme recruited. There is a recruitment of an enzyme which we called DNA helicase. DNA helicase. Helicase is the enzyme which is basically responsible for breakage of hydrogen bond between the two strands of the DNA. So let us say these are the two strands of the DNA. What DNA helicase will do? DNA helicase will break apart these two strands of the DNA by breaking the hydrogen bonds. So at least we can start the process of the DNA replication. So in the next point, there will be a replication fork. This is what we call replication fork, and replication fork is basically formed by DNA helicase. So this is our helicase. This is our helicase here. And this helicase have broken down these hydrogen bond between these two strands. So we have broke these two strands hydrogen bonding. So the helicase is moving in this direction, and it is basically breaking hydrogen bond at a much faster rate. So now these two strands means now the DNA polymerase at least can come and start the process of the DNA replication. You know the DNA polymerase. DNA polymerase means an enzyme which is going to replicate, the DNA. But there is a problem. As DNA helicase, this enzyme here is moving in this direction. The other part of the DNA, which is already apart, because of their affinity, will start moving towards each other, and again they will form hydrogen bonding. तो जैसे ही हेलिकेज आगे जा रहा है, वैसे वैसे पीछे की जो स्ट्रैंड है वो क्या करना स्टार्ट कर देंगी, वापस से हाइड्रोजन बॉन्डिंग करना स्टार्ट कर देगी. उनको भी हमको स्टॉप करना है. फॉर स्टॉपिंग दिस we have an enzyme. And this enzyme is what we called single-stranded binding protein. So this single-stranded binding protein binds to these single strands of DNA which was basically separated by DNA helicase. This enzyme, single-stranded binding protein, prevents the hydrogen bonding between already apart single strands of the DNA. तो ये जो बेसिकली सिंगल स्टैंड बाइंडिंग प्रोटीन है, दिस इज द सिंगल स्टैंड बाइंडिंग प्रोटीन, ये वाला काम नहीं होने देगा. दैट मीन्स दिस DNA will not come together. Otherwise there would be a lot of problem. This is the 5' DNA, this is the 3' DNA, this is the 3' DNA, this is, remember the ends are very important. There is another problem. As the DNA helicase is basically moving, DNA is not like this. DNA is not this ladder-like structure. DNA is coiled. DNA is basically coiled. And if you are basically breaking the hydrogen bond, the rest of the DNA will be super coiled. There will be the number of coiling will be increased in the rest of the DNA. So this part of the DNA gets super coiled. This part of the DNA gets super coiled. So there is another problem. पहली प्रॉब्लम क्या थी कि जैसे जैसे हेलिकेज आगे बढ़ रहा है, दिस इज ओवर हेलिकेज जैसे जैसे आगे बढ़ रहा है, पीछे का डीएनए वापस से हाइड्रोजन बॉन्डिंग कर लेगा. उसके लिए हमने कौन से एंजाइम का रिक्रूटमेंट किया? सिंगल स्टैंडर्ड बाइंडिंग प्रोटीन. दूसरी प्रॉब्लम क्या है कि जैसे जैसे हेलिकेज आगे मूव कर रहा है वैसे वैसे देयर इज सुपर कोइलिंग इन द रेस्ट ऑफ द डीएनए, कोयल का नंबर इंक्रीज हो जाएगा. दिस इज व्हाट वी कॉल्ड सुपर कोइलिंग. दिस सुपर कोइलिंग cause that destability in the DNA, can cause destability in the DNA. And if there is destability in the DNA, there will be no DNA replication. Whole DNA will get destabilized. So we have to prevent this destability, which is basically caused by DNA helicase. So in order to prevent this supercoiling, we have an enzyme. There is another recruitment of enzyme. There is another recruitment, and this recruitment is topoisomerase. So there is another recruitment. So to prevent this supercoiling, there is another enzyme. This enzyme is what we called topoisomerase. What topoisomerase do is, this causes a nick in DNA. This is the topoisomerase here, the green color part. This topoisomerase basically causes a nick in the DNA. So if there is a nick means there is a small cut. Nick doesn't mean we are removing the base pair from the DNA. Nick is basically what we call breakage in the phosphodiester bond. तो जो backbone है उस bond को तुमने break down कर दिया with the help of topoisomerase. That means that tension is relieved. एक छोटा सा break किया in the phosphodiester bond, and due to that break क्या हुआ, tension जो थी, जो super coiling थी, वो क्या हो गई, remove हो गई. So this is the role of topoisomerase. Now the DNA is available for replication process. एंजाइम आ सकता है डीएनए पॉलीमरेज यहाँ पे, एंड डीएनए पॉलीमरेज आके क्या कर सकता है, आराम से रेप्लिकेशन की प्रोसेस कर सकता है, बिकॉज देयर इज नो टेंशन. These two strands जो हमने पहले से अपार्ट कर रखे थे, they are already moved apart with the single strand binding protein. तो these are open. ये बोल रहे हैं कि अब we are ready for DNA replication process. This part here is what we called replication fork. This part here is what we called replication fork. So now there is the role of DNA polymerase. We will discuss the role of DNA polymerase in the next lecture. I hope you liked the video. Thanks for watching the video. Please don't forget to hit like and subscribe before you leave. Thank you and see you soon in the next video.